Hi, good morning. How are you all? In this slide, we will know about mechanism of muscle contraction. Muscle contraction is physiochemical mechanism. Physical changes are explained by sliding filament theory. Sliding filament model. Sliding filament model. The sliding filament model of muscle contraction was put forth by Huxley, A.F. Huxley and Hansen in 1954. According to their theory of contraction of sarcomere depends on the presence of two set of filaments. During muscle contraction, thin filament slide over the thick filament, resulting in shortening of sarcomere. A series of events occur during this process. Those series is stimulation of muscles, contraction of muscles, relaxation of muscle. Stimulation, contraction and relaxation. First we will see one by one. Stimulation of muscle. Stimulation of muscle when a nerve impulse reaches the neuromuscular junction, the sarcolemma of that muscle gets depolarized. The depolarization extends up to T-tubule and cisternae of sarcoplasmic reticulum. Hence, calcium ions release into the sarcoplasm from cisternae of sarcoplasmic reticulum. These calcium ions diffuse into the thin filament. So here 3 points in stimulation of muscles. One is nerve impulses reaches to the neuromuscular junction, then the sarcolemma of muscle cell gets depolarized. Depolarization extend up to the T tubules and cisternae of sarcoplasmic reticulum. Hence, calcium ions release into the sarcoplasm from cisternae of sarcoplasmic reticulum. These calcium ions diffuse into the thin filament. Next one is contraction of muscles. Contraction of muscles. Troponin has three subunits: TNC, TNT, and TNI. Calcium attach to the TNC during muscle contraction. Tropomyosin attaches to the TNT. Calcium to the TNC. Tropomyosin attaches to the TNT. So let us see. First one is the calcium ions bind to the TNC of troponin protein in the thin filament. Next, binding of calcium ions to the TNC shifts the tropomyosin complex, so the active site are exposed in thin filament. The increase in the concentration of calcium ions also causes the conformational changes in the myosin that permit the binding of myosin in thin filament at active site. In the absence of ATP, myosin head binds to the thin filament at active site tightly, forming actomyosin complex. When ATP binds to the pockets of myosin head, this bond weakens. ATP as present in the head of myosin filament hydrolyzes the ATP. Hydrolyzed ATP causes conformational changes in the head of myosin. So the head of myosin moves a new position and recombines with another active site of thin filament. Next, actin myosin complex at active site release inorganic phosphorus. It swings the cross beads towards the H zone. It is called power stroke. The thin filament slide over the myosin filament. This power stroke is followed by the release of ADP, the conformation of myosin, which is with a pocket of ATP and tightly bound to the active site, is restored. This is all changes under contraction of muscles. Next, relaxation of muscles. Relaxation. Calcium ions detach from the thin filament and diffuse towards sarcoplasmic reticulum. Troponin permits the tropomyosin over the active site of thin filament. Myosin actin cross bridge breaks. This is the relaxation of muscles. This is some information about muscle mechanism of muscle contraction. Okay. So in this slide filament theory, thin filament slide over thick filament, resulting shortening of sarcomere. Okay, thank you.